Okay, so Fernando will give his second lecture.
So, good afternoon. So we will resume where we left it yesterday, basically, which was to trying to understand how genes are expressed in different places and at different times during development. So different parts of the animal, as it develops, will become different types of cells and will do different things. So if you remember, yesterday we ended up by talking about cis regulatory elements, pieces of DNA that usually are not non-coding, where transcription factors bind. Okay? And depending on the binding of these transcription factors, the gene will be expressed or not in the place where these transcription factors are expressed. So these sequences are actually performing a computation. Right? So they will be binding transcription factors if they are present, and the result of that binding will be the activation of the transcription of a protein coding region or a RNA coding region. So this is important because the transcription factors typically bind to sequences that are specific, like this one. So here you have a little stretch of this enhancer element, and when we read it, we will see that in these positions, the transcription factor prefers binding a G, and followed by an A, followed by an A, followed by a C. Right? That's the preferred sequence that this particular transcription factor that regulates the transcription of the gene even skipped in this stripe called cruple likes to bind. And that's basically because these transcription factors have domains in their protein that recognize specific shapes in the DNA, and these shapes are determined by these sequences. There are one thing that you should notice here, is that this cruple transcription factor, it doesn't matter which transcription factor it is, binds preferentially to a short sequence, right? GAAC, say. So how frequent is this sequence in the genome? So you can calculate that very easily. Here G is one of the four bases, so this would be 1/4 and 1/4 of A, and 1/4 of A, and 1/4 of C, which is 1 in every 256, I don't know, bases. So remember that a genome is millions of bases in length, right? So cruple on its own should bind thousands of places in the genome. That would make this transcription factor very promiscuous, right? So many genes should have cruple binding sites nearby. But this is not how it works in real life. The activation of an enhancer does not depend on a single transcription factor. It depends on a combination of transcription factors. So now imagine that instead of looking only at cruple binding sites, we had here a number of these red boxes, which might actually represent binding sites for different transcription factors. Now you multiply the probability of finding them close to one another in the genome, and that the probability is much, much reduced, right? So this enhancer will only be active, and therefore driving the expression of even skipped when a particular combination of transcription factors are present. And that gives this exquisite specificity, right? So these binding factors not only measure the presence or absence of transcription factors, they can also compute concentrations. So it might be that cruple here binds only when cruple is at high concentration, but there might be some other places where this site is slightly different, and cruple would bind with a higher affinity, so it would bind where cruple is at lower concentrations, and sometimes you have cooperativity of transcription factor binding sites. So two transcription factors will bind and activate an enhancer only when the two are present, following some sort of an AND logic. Only when transcription factor A is present and transcription factor B is present, then the enhancer would be active, right? So this is important. So enhancers basically are able to read transcription factor concentrations. And we mentioned yesterday that the transcription factors that a cell expresses at any given time depend on the cell's history. So if you come from a mesoderm and you're a muscle cell, you keep track of that you derive from the mesoderm because you're expressing certain transcription factors. And also depends, the transcription factor collection will depend also on signals coming from other cells, right? So we this morning heard about induction, right? So there are certain molecules, if you are close to those cells sending you these molecules, you will activate a signal transduction cascade that will end up activating a transcription factor, and you will have that transcription factor as opposed as some other cells which are far away from the inducing cells, okay? Right. So you saw this yesterday, and these are major characteristics of these enhancer elements. So basically just simply to reinforce the idea that when cells are changing the concentration of proteins within their life, they are doing that through the action of cis regulatory elements. Okay, that's really, really important. So I decided to throw in a number of slides showing you some experimental ways of finding these cis regulatory elements, these enhancers and repressor sequences that act regulating the activation of promoters and the transcription of genes. One is like in the old-fashioned way. One just simply bangs the animal's genome and looks for phenotypes. And in this particular case is one phenotype that we've been seeing once and again. It's called bithorax phenotype. A bithorax phenotype is a phenotype that transforms partially the haltear, this balancing organ, into the likeness of a wing. Only the anterior half, okay, this is the bithorax mutation. Only the anterior half of the haltear has been transformed. And how do you guess that this mutation affects a regulatory region rather than a protein coding region, for example? Because you might have another mutation that actually destroys the protein coding region. And when you combine the two mutations, you will find that the mutation is like this one, not like the total loss of function of the protein function. Okay? So this is not really a total loss of function. But yeah, it should be like that. So the bithorax mutation produces a phenotype that is smaller, is a subset of phenotypes of the total loss of function of the gene. So that might tell you that you are hitting here one of these control elements. That regulates the expression of this gene that is required to make the haltears, otherwise they are transformed totally into wings, and is required in the anterior region of the haltear. The posterior region of the haltear is fine, this mutation, all right? So the other way of looking at what's the activity of whether you have a sequence that has enhancer activity is exactly the way I've been using so far. You take a piece of DNA, right, which is this one, and link it upstream of a promoter, which is this little arrow here, and a transcription unit that in this particular case is just simply going to be a reporter of transcription. Here you can put any protein coding gene that is easy for you to recognize. So for example, you can use LAC-Z, which is beta-galactosidase, the gene that Jacob and Monod actually studied, just because we have an easy way of detecting where LAC-Z is produced. But it could be perfectly GFP, a green fluorescent protein. So wherever you have green fluorescent protein expressed is because the enhancer is working. So these constructs work because this promoter, where is the landing site for the RNA polymerase, remember that for a gene to be expressed, it needs to be transcribed, and transcription is a business carried out by a macromolecular complex of many proteins called RNA polymerase. RNA polymerase also detects specific sequences in the promoter region, okay? But if there's no enhancer, this particular promoter, even though it can bind RNA polymerase, won't drive any expression, right? So it would remain silent unless it has an enhancer. And the enhancer will be active only in those places where the transcription factors it recognizes are present. In this particular case, in here and in here. So with this assay, this is basically a transgenic assay in which this construct now has been transduced into the genome of every single cell of the embryo. So all the cells contain this transgene. Only these cells in here and in here will be activating transcription of the reporter gene, and therefore this piece of DNA is recognizing something very specific about what goes on in this band and in these cells in here. That's the idea of how you, for example, test introngenesis, the regulatory activity of a piece of DNA that has cognate enhancer or functions. All right, the other thing and actually, yes?
So you said that bithorax mutation primarily affects the volunteers and makes them big.
Yes.
Right? But why is there a duplication of thorax region accompanied alongside?
Okay, that's because there's a misdescription of a phenotype. So the bithorax mutation actually duplicates the thorax, not only the wing. When we talk about the thorax of a fly, it contains both the trunk part and the wing. So bithorax mutations actually duplicate the whole structure. The whole structure actually comes from the same group of cells.
Are there instead of imagining like volunteers change into wings, are there another set of volunteers in the above?
No. So imagine that you had, we will come to that in the next slide, in the next lecture, but just simply to give you an idea. So these would be the second thoracic segment. This one, it contains the notum, large, and the wings, large. And here you have the third thoracic segment, that is a strip of cells, much narrower, and the whole tears. So even though you cannot see this in this micrograph, the bithorax mutation here transforms partially the whole tear into a wing, but also this trunk part, okay? So it affects the whole segment, right? So it's not just a transformation of whole tears into wings, it's a transformation of this whole segment, the whole third thoracic segment into second thoracic segment. So the other way of that people have been using to identify potential regulatory regions is by genome comparison. So this is reasoning that molecular evolutionary biologists use all the time. So if you compare two sequences, if they are not that far away apart, you will find that sequences that are functional will tend to be more conserved than sequences that are non-functional. So, okay, imagine that you now align two genomes and you find two stretches of conservation. The first stretch affects a region that is coding. No surprise there, that's going to be a homologous gene, right? Do you have a, no? Then let's go to the other stretch. The other stretch is non-coding. So why is it that this non-coding piece of DNA is more conserved than the background sequence? So the idea would be this sequence is doing something, and therefore you cannot vary it too much. So this is the criterion used, for example, in this type of genome analysis. What you have here is an alignment of a piece of DNA, piece of the genome, of a mouse compared to the chicken, the frog, and the zebrafish. And you have to trust me, this is a non-coding region, right? So, and this is human. So when you compare that, you will see that in this stretch of DNA, only this is very conserved in vertebrates, right? And this is non-coding. What you do now is exactly what I've told you would do with the even skip stripe. You would clone this piece of DNA upstream of a neutral promoter, driving the expression of LAC-C. Now you generate transgenic mice, in this particular case, and then you see that this piece of DNA that you've identified, conserved in all these animals, is expressed in the developing limbs of a mouse, right? You could do the converse experiment, is to take, for example, the mouse sequence and transform with it in the same manner, with the same constructs, a zebrafish. Most likely you will see that this sequence, even though it comes from mice, it will be expressed in the developing fins, excuse me, of zebrafish, okay? Yes?
So those genes are the ones who make the protein. Those are coding genes or not?
No, these sequences, I stress that, these sequences are non-coding in this particular example. So these are sequences that are evolutionarily conserved, but are non-coding. So that's one criterion that people have used to identify regulatory regions, non-coding regions, but regulatory regions with enhancer activity, for example.
Further process of limb development will carry on as.
Okay, in this particular case, we are not doing any mutation analysis. Okay, in this case, we are not mutating this region. But nowadays, for example, if you use certain techniques, you would be able to specifically, for example, in the mouse, remove this sequence specifically from the mouse genome. And if you bring that deletion to homozygosity, so the two copies, the two alleles carry the mutation, then you can ask, okay, what does the removal of that enhancer do to limb development? So when looking at this, you would imagine that it's going to have an effect only in limbs and nothing else around it. Otherwise, it would be a surprise. But nevertheless, this is the way you would do to follow up on identifying some non-coding region with enhancer activity. You want to analyze the function. But there's more. There's more in the chromosomes than DNA, right? So biologists are familiar with this, but perhaps this is not so well known by physicists. Is that DNA, even it's a long polymer. It's a long polymer. It tends to be like, it doesn't like to be bent on its own. So for compaction, and some other things, the DNA goes around complexes of proteins called histones, right? And once they are wrapped around the histones, these histones and this material can further condense. So that, in extreme cases, is what you see during cell division. When the cells are dividing their genetic material, the chromosomes are compacted and condensed to a maximum to allow for an easy dispatch into the daughter cells, right? But there's something really interesting about these histones. The histones are not just simply sitting there allowing the DNA to be packed, but these histones can be modified. And the modifications, these are covalent modifications, like, for example, adding acetyl groups or phosphate groups to specific residues on specific histones here. And depending on the code, this piece of DNA will be recognized differently by transcription factors. Okay. So, for example, the presence of certain methylated residues will be an indicative that this piece of DNA, which is non-coding, is actively acting as an enhancer, as a cis-regulatory element. So now that we know a little bit about the coding of these histone modifications, now we can get profiles along the genome where we can detect these different marks. Okay, so this is typically what you will see on a piece of chromosome. So here is a piece of the genome where there are two genes, SP7, SP1, it doesn't really matter. But the important thing is that there will be regions that are enriched in histones with a particular mark, while there are some other regions where these the histones are not marked specifically. And by looking at the expression or presence, of certain marks, we can define regions which are likely to be enhancers. Right? So now we can have a look at the whole genome and identify those regions that are actively working as enhancers or as regulatory regions. No, this is the same organism. It might be one cell type. It might be a chunk of the embryo. But here what you have is, so these are different marks on the chromosome. So this methylation, a methylation in a particular histone here. This is another type of modification, which is an acetyl group bound. This is the presence of a particular transcription factor, which has a very important structural role, et cetera, et cetera. So you will see that there are places where these things coincide, which are For example, in this particular case, it's the promoter of the gene. There are some other places where these marks coincide and they are not at the promoter, so this region of the chromatin is likely to be acting as an enhancer. Right. Sure.
How does this non-random marking on the
That's a very interesting question. I will give you a glimpse of how it happens, but Dr. Laga most likely will explain that in great detail, actually. But yeah, it's a clear demarcation, and actually it depends on how you zoom in or zoom out on the chromatin. You will be able to actually detect large blocks of the chromatin that are closed for business, and some others which will be open for business. So you can classify the chromosomes like painting the chromosome in regions that are mostly closed, mostly active as enhancers, etc. Okay, so there are different chromatin domains. There was the question of how you start marking those. So that's a really interesting thing. It seems that there are some transcription factors that are able to recognize an enhancer without the need of it being marked. Okay, those are called pioneering transcription factors. So once a pioneering transcription factor gets there, it somehow recruits other transcription factors. And then these other transcription factors will modify the chromatin. And that chromatin modifications will allow these transcription factors to recruit further transcription. In this case, would be molecules that will bring that piece of DNA close to the RNA polymerase and then push it in a way, so transcription will start. So there are pioneer transcription factors, and there are some other transcription factors that also come afterwards. But importantly, you might have a pioneer transcription factor, and yet the enhancer is not working. Why? This pioneer factor has marked the enhancer, but it requires the binding of other transcription factors for the enhancer to be active. Okay. Yes, that's the linear sequence of the DNA.
So, with the modification that we done the chromatin, the histone protein.
Yes. Yes.
So, does it mean that the histone protein is always in the same location and the
Okay, that's a good point. I mean, the resolution of this thing here is perhaps 100 base pairs or 200 base pairs. So you might have the nucleosomes, the histones, not localized exactly in the same position. Although, because of the way the length of a DNA goes around these histones, there will be a certain specific pattern. Yeah, like a ladder. Okay. Okay, so I'm giving you a little bit of a biochemistry here because you might be interested in knowing how you produce this sort of data. You produce this sort of data because you can actually use antibodies that will recognize the different types of modifications on the histones. Okay, so the procedure works as follows. Imagine that you have one of these histones, which we will call histone star, and you have an antibody that recognizes specifically the star form of the histone. First thing you do is you covalently link the histone to the DNA. Okay? Remember that these guys are just simply, the DNA is just simply initially wrapped around the histone. But if you start extracting biochemically things, you will dissociate the proteins from DNA. So the first thing you do is you cross-link covalently the protein to the DNA. Okay? Now you have the protein. Some of them will be stars, some of them won't be stars. I mean, now you would like to get the sequences that are bound by the star form of the histone. So you would throw in your antibody would bind to all the stars, not to the others, and then you pull from the antibody. Of course, pulling from a long strand of DNA is very difficult, it's very viscous. What you do first, before anything, is you chop the DNA into little pieces, right? So then, once you have your antibodies bound, you will pull, using some biochemical techniques, you will recover those pieces of DNA that are bound to histones that are star modified because they are bound by the antibody, okay? And you discard the rest, the unbound. And then you sequence every bit of DNA that you've pulled out, right? So of course, the sequences that are initially associated with the star histone will be very enriched in your sequencing reactions. These will be small sequences. And now what you do is just simply project those sequences, align those sequences into the genome. And what you get is regions where there's no binding of force, because these they weren't associated with the star histone, but some others where you recover many. And each of these little things that you have here piling up are small sequences that come because they've been associated with the star histone. You get the idea? Say that again?
The other sequences which they are not bounded by histone?
You discard them. So you pull the ones that are bound, and the others go into the supernatant. Forget about them. Okay? You sequence what you've retained, and then you project them onto the genome. You align it to the genome, so you localize where they come from originally. And this is how these, you have to do some statistics, but this is how you get these peaks that we were localizing on the DNA. You not only get one, you get all the places where this particular histone modification is particularly enriched. Okay? And if, for example, this star histone is associated with enhancers, then you are going to detect the regions on the DNA, on this chromosome, or in this genome that are enhancers. Yes? That's a technicality. So when you sequence these things, you sequence from one, remember that the DNA are two strands. Yeah. So then you will have a way of sequencing from one end and from the other end. So then, even though it's the same sequence, the sequence coming from here will be on the right, and the sequence coming from here will be on the left. So then you have to project them onto that, and then you have to make some assumptions that because there is some separation between the left and the right halves, what you are detecting is a bit like the summation of the two. So, yes. Yeah, but yeah. So this is going to be the left, and this is going to be the right part. And this is the DNA. So these are complementary. So if you were able to actually sequence the whole thing, you would map exactly to the same position. But imagine that the sequence that you get are these. Excuse me. And these. And now you map them onto the genome. So you will have, like, right? For a sequence, you will get two sequences, which are not associated. Then you will get some other like that. Depending on how large the piece of DNA, which is a bit random, you will get something like that. And then something like this. And then when you sum up this whole thing, you will get something like that. But this is part of the same molecule, right? This is the same read. So that's how. No, it's not meaningful. It's just simply that it derives just simply because of the way you sequence. Okay. Yeah, there's nothing biologically relevant in the bimodal distribution. Just simply it's derived from the technique. Okay. No, it's not a problem. So what you get is you get a complex that is antibody recognizing the histone, and since the DNA is bound to it, if you pull from the antibody, you get the DNA sequence. You get the DNA, excuse me. So then you get that, you extract the DNA from there. Now you free it from the protein and from the antibody. You get little strands of DNA floating around. Those are the DNA pieces that in the genome of the cells of interest are preferentially bound to this marked histone, and then you sequence everything. Yeah, it cross-links, but then you can revert the cross-linking. And okay, that involves quite a lot of biochemistry, right? So this technique is nice, but it involves quite a lot of biochemistry. But very recently, Buonrostro and collaborators invented a new technique, which is great. So one of the things that happen when a region of DNA is acting as an enhancer is that many of the histones actually are displaced to the sides, right? So they are bound by transcription factors which are bulky, but on average, the DNA is more naked than if the DNA is not bound to transcription factors. Weird to imagine, but imagine that histones are small proteins. They are bound to the DNA uniformly. But if a transcription factor comes in, it will just simply make room for itself. It will bind to very few places, but because it's bulky and changes the structure of the DNA, it won't allow the histones to be bound again. So it's like a bit of a naked DNA. So being naked, the DNA, it becomes more accessible to some other DNA molecules. In this particular case, what Buonrostro and collaborators did was to use one enzyme that is able to detect the places where the DNA is more naked and makes breaks there, right? If you add that to some sequencing tricks, you will mark with these little sequences those regions preferentially that are opened in the genome, okay? So in one reaction, to chromatin, you add the TN5 transposase, and that will mark immediately those regions that are open for business, like enhancers and some other regions. And then you do exactly the same thing. You take away the regions that are bound to histones. Okay, you remove them, and now you keep only those regions that are naked and marked by the transposase. Now you sequence everything, and then you come up with those regions where transcription factors have displaced away the nucleosomes, the enhancers. And that's really, really fast. That's a biochemical procedure. This is really fast. You do that in a few hours, then you produce a library for sequencing, you send it for sequencing, and you get the sequence back, and then you can project it on the genome and identify, for example, places like these ones. This technique is called ATAC sequencing, and this is a typical profile around the gene in Drosophila, which is called Cineoculis. It doesn't really matter, and you can see here there are places that are kind of open for business, and now you get this region here that is open, and then you do the transgenesis assay. Now in this particular case, you have this piece of DNA hooked up with GFP. You generate transgenics and look where the GFP is produced and is expressed. You have to tell me to trust me. You will see more of this in a next week. But this is the I primordium is expressed specifically in some eye cells. So with that, you've identified a piece of DNA with important regulatory function of the gene in order to make an eye. And you can see here, for example, other marks and of other techniques, which actually are very similar. So now, and this to wrap up the idea of how molecularly genes are characterized. They're characterized by a complex mixture of DNA segments. You have DNA segments that are transcribed. Some of these transcribed regions are going to go into a protein. Some others are going to be spliced out. There's some combinatorial in this splicing, so you can generate different forms of the protein by including these or these exons. So the exons are the segments of the DNA that are going to go into proteins. The introns are the sequences that are in between. Of course, you're going to have promoters, where the RNA polymerase sits. There are some other additional sequences that are going to go into the messenger RNA, which are really important in controlling the stability of the RNA. They are important in regulating the speed at which, or the rate at which, they are translated into proteins in the ribosomes. And they are also targets of those microRNAs I mentioned yesterday. So those are places where microRNAs, if they find the target in this region on the RNA, will go and bind and destroy the RNA, regulating the concentration of this specific RNA. And then you have regulatory regions of the ones I've just described. And they can be very modular, very specific. One gene with a very complex pattern of expression, for example, one that is expressed in the brain, in the limbs, and in the liver, might have specific enhancers for each of these tissues, okay? So, and they can, and this is something really important and really remarkable, is that these pieces of DNA, these enhancers, can act at really long distances. So here you have the case of one important developmental gene, which is called sonic hedgehog, that is a secreted signaling molecule, and has, yes, that funny name, sonic. Sonic is expressed in the limbs. Okay, that's the normal expression pattern of sonic in the limb bud of a mouse or your hand when you were a baby, right? When you were a very tiny baby. Well, this pattern of expression is actually controlled by a piece of DNA that is located 1 megabase away from the promoter sonic hedgehog. And actually, this piece of DNA is embedded in another transcription unit, this of the LMPR1 gene. So when people start like mapping mutations associated with some human diseases, they thought that this piece of DNA should be actually regulating this DNA, this gene. But in fact, it's regulating sonic hedgehog here. So when this piece of DNA in a mouse is removed, what happens is that sonic hedgehog is no longer expressed in this posterior bit of the limb bud. And these mice do not have a hand or a foot. Right? They lack all the distal part of the limb that corresponds to the autopod, the hand or the foot. And interestingly, there are some mutations in this piece of regulatory DNA in humans that do exactly the opposite. They hyperactivate the transcription of hedgehog. Actually, they drive the expression of hedgehog in ectopic locations. And they give rise to these sort of polydactylia syndromes. These are human syndromes that are associated with mutations in this enhancer that cause, when these mutations have been introduced in the mice, they produce an ectopic source of sonic hedgehog. So that disrupts totally the patterning of fingers in the limb. Yes. Yeah, it might be possible, but none has been detected here. Exactly, we know that for sure. So I'm not showing you the details, but we know that physically this piece of DNA contacts the promoter of this gene, and not, for example, the promoter of this, and ignores the other one. Yes, exactly. I will tell you why this happens in just one slide. But going back to this morning talk, where we were discussing multicellularity, the group of Ignacio Ruiz Trillo in Barcelona has been studying Capsaspora, right? One of these semi-multicellular organisms, right? That were described this morning. So they've analyzed the genome of Capsaspora in great detail, and they've been looking for protein coding regions, and the Capsaspora contains a great collection of genes, basically corresponding to all the genes that you need to do a multicellular organism. So why is it that they are not multicellular? Basically, what they found is that although they have the potential to do it, they lack, by and large, these long-acting cis regulatory elements. They haven't invented enough sequences capable of regulating in space and time the genes that they already have. Right? So multicellular evolution required not that much of an evolution of new proteins, but of instructions that can regulate where and when these genes are expressed. And this is evolution of the cis regulatory elements. And further, so chromatin is in order for it to be properly regulated, as your fellow student just mentioned. So why is it that this sequence is regulating the gene 1 megabase away and is ignoring these two other transcription units? This one is more difficult to explain. But so imagine that for whatever the reason, there would be like some sort of isolation of this promoter from this other promoter. And there would be a barrier separating this enhancer from this promoter, but not from that other. This is how actually chromatin is organized in real life. So there are regions that will bend and would get in closer proximity. So all this piece of DNA in between these borders will be in close proximity. So everything that is within here will be prone to interact, while any other piece of DNA that is outside these borders won't be able to interact with them, or not that efficiently, although they might actually be interacting within them. Okay, these regions of the DNA are called topologically associated domains, right? So this is a way of confining long-range regulation to specific segments within the chromatin. Right? So this is higher level organization of the chromatin.
For example, on one chromosome that enhance some gene of the other.
Well, okay, I don't think that there are examples of that. Not too many, at least. In Drosophila, there's a phenomenon called transvection, in which an enhancer of a gene located on one of the two chromosomes might actually be contacting the promoter of the same gene on the other chromosome, the homologous gene, so in two alleles. Right, this is called transvection, and it might happen in some other organisms. But by and large, since gene regulation is confined to topologically associated domains, topologically associated domains haven't been seen to comprise more than one chromosome. So I think that nowadays the consensus would be no. Enhancers regulate promoters that are located within a single TAG, and TAGs do not span more than one chromosome. No. I told you that if you have a gene expressed in many different places in the body, you make this complex pattern of expression by having different enhancers specific for each of the regions.
That means that and this thing is similar for all the members of the species, right?
Right.
It's for all the members.
Yes.
That means this folding must not be random. It's very clearly organized.
Yes.
It's not a random folding at all.
Exactly. It's not random at all. So the borders of these topologically associated domains are marked by sequences that are bound by very specific and peculiar transcription factors. Okay? Yeah, so these places across the genome, along the genome, are marked very specifically by sequences that are actually conserved. So yes, you're absolutely right.
Chromosome means one, there are two chromosomes per.
Okay, yes?
I don't know the chromosome.
Yes?
So, for every one single chromosome, you have.
Yeah, exactly. So you might have, this would be one chromosome. We have a number of pairs of chromosomes. So this would be one pair of chromosomes. So this would be one tag. Imagine this chromosome 2, and this is the other chromosome 2, you will have the same thing here. Right? And these are identical, because basically you might have mutations, of course, that affect the formation of this TAT, and you might have one chromosome doing like that and some other not doing it. That might have nasty consequences on how the animal develops, because regulation is going to be altered. But anyway. So, okay. So finally, we've come up to some sort of summary slide in which, as development proceeds, every cell, depending on its position, will be expressing different sets of transcription factor during time. So, for example, this group of cells here, okay, you can define them by some sort of vector, and that vector contains concentrations for each of the proteins encoded in the genome. Right? That would be what characterizes cell state. It might actually be a bit more complicated than that. It might be characterized by a distribution, right? So you would have G1 would be actually a distribution of values. G2, which is a gene product, would be another distribution of values. But this is how you would characterize a cell. So then these cells will grow, and they will keep on subdividing the tissues into different regions. And here you have, this is the primordium of the thorax that we've been discussing quite a lot. This is an epithelium. And you will see that there are regions which are red, and next to regions that are blue, and some, so here you also have this system of coordinates. We've been talking about coordinates in the embryo, dorsoventral, anterior, posterior, but you keep subdividing these groups of cells into smaller and smaller regions, right? So you have that these cells that come from this particular stripe, now they are going to be subdivided into something that is going to be the dorsal, the ventral region, the posterior region, the anterior region, etc. And finally, you have the final structure. So from this piece of, from a few cells in here, you have this primordium that will give rise to all these cells, right? Including cells that are going to make the wing, sensory cells there, like cells that are going to make the carapace of the animal, etc. So, therefore, through development, cells, as they divide, they seem to be making decisions on where to go in a potential landscape, right? So that's a very old concept. It's a metaphor that was coined by Conrad Warrington, who was extremely creative scientist. So this is a way of seeing development. So you have initially cells that are really multipotent, and at some developmental transition point, they either go this way or this way. Right? And if they go this way, they only can then go to here or here, right? Because their development is, or their decisions are being restricted, they are canalized. Again, he called these sort of valleys creoles, so he invented all sorts of words. But the interesting thing is that he gave some sort of biological meaning to this sort of landscape, is that the shape of this landscape was modified by the genes, so different alleles of genes, the action of these genes, in very complex ways, right, would be pulling from the surface and shaping it, okay, as development proceeds, like describing the shapes. And therefore, controlling where a cell is going to land, okay, which sort of cell this is going to be. Sure. We'll go back to that. I mean, he imagined genes as these pegs in here, but these connections actually, you might imagine also being influenced by many other things. Okay, genes were definitely a component to that. But actually, the action of genes and even the expression of genes depends on the environment. So this becomes much more complicated and dynamic than actually how this representation can convey. So nowadays we are able to, by sequencing the expression of genes in single cells. Now what you can do is to get an embryo, dissociate the embryo into single cells, and then get every single cell and ask, what is the value of that vector I mentioned? So what's the expression you have for every single gene of your genome? And by doing so, people have been able to reconstruct these developmental trajectories of cells that Wellington depicted. So here you have this cell here, which would be the multipotent, or basically the zygote, and it might, these guys have ordered, in a way, potential paths that will lead from this cell to many different cell types that are present in this particular case. It was like a late larva, a zebrafish larva. So, so now we are getting to a bit about the dynamics. So the epigenetic landscape of Wellington, that valleys that Wellington represented, that the genes acting on them are not acting instantaneously, right? Instantaneously. They're just, this is a progress that goes on as the cell develops. So we need to understand what controls the dynamic of gene expression along development. So what is the actual machinery that controls the differential expression of genes in different places as time passes, right? And here, I think that one of the scientists who advanced this field very much was Eric Davidson. Eric Davidson worked with sea urchin, but it doesn't really matter. Sea urchins have embryos as any multicellular organism. And he started like connecting the expression of genes with the actions of these genes on other genes. So for example, in this particular example, it doesn't really matter, it's for the specification of a particular cell type. The work from his lab was able to find out that it was a certain patterning process was controlled by a network of interactions involving two transcription factors. These two guys are transcription factors, right? So this, for example, OTX is able to activate BLIMP1, and by a signaling molecule that allows the communication between cells, which is Wnt8, that in the end, when a cell receives Wnt8, it will actually act on a Wnt-specific transcription factor. So here you have, in a nutshell, what I mentioned before. So cells change their gene expression and therefore their fate because there's connectivity between transcription factors acting on some other transcription factors and signaling molecules that come from outside, from the cells nearby. And importantly, remember, this is just simply the structure of the logical circuit. Since these guys are transcription factors, they themselves control many other genes. So when we say that OTX is expressed and BLIMP1 is induced, we are also obviating the fact that OTX is controlling many other transcription factors, many other genes. So when OTX is expressed, these cells are expressing a particular set of genes. When OTX has induced BLIMP1, now because it has this transcription factor, new genes will be regulated. Okay? And this now has, it's like a big chemical reaction, which has also a temporal component to it. So first you have to have OTX. So once you have OTX, later OTX can bind BLIMP, can induce the production of BLIMP1B, and that will act now on this other signaling molecule, and so on and so forth. You will see the pattern of genes changing over time. Right? It's not an instantaneous operation. It happens with some delays. And because they are chemical reactions, and this is important, they impose a developmental time themselves. So the delays built in here are the delays that actually mark the pace of development. So you cannot go faster than these chemical reactions. Okay, so that's important as well. And basically, it's just simply a restatement of what might happen when one tries to understand the process globally as a dynamical system. Is that the regulatory state, which is the set of transcription factors that the cell has because of its history and signals, is somehow integrated at the level of cis-regulatory elements, right? And these changes then are amplified, are processed in the genome through cis regulatory elements. And those changes in gene expression are the ones that we detect as cells differentiate. And in a way, these terminal states are some sort of attractors that the system can reach in a stable manner. All right, and here I'm just simply bringing a bit of work we did a number of years ago, which is some sort of cis-regulatory element paleontology, right? So to tell you how the study of gene expression regulation can tell you about very old events in the past. So what we did was to compare the sequence, the non-coding sequence, across many different genomes, okay? And we found just a few sequences which were non-coding that were conserved in a huge range of animals, okay, from humans to Nematostella, which is basically a polyp. Right? So this sequence, you can see it there, like this little peak here is a bit of sequence that is conserved from humans to Nematostella. There are very few of those. So these are like very strange sequences. But the interesting thing is that when we took this sequence from humans, or from Amphioxus, or from sea urchin, and generated transgenics with them to ask, where you very weird sequence is acting as an enhancer, we found that all these sequences drive expression in undifferentiated cells within the brain. We did that experiment also by taking some of these sequences and generating transgenics in sea urchin. And in the sea urchin, that piece of DNA is also expressed in early neurons. Further, we did an experiment with Drosophila. Drosophila doesn't have this sequence. Actually, it doesn't have this gene. Right? It doesn't have this gene. But we asked, okay, are there any cells in Drosophila that have the same regulatory state? Which means that there will be some cells in which this enhancer will be activated. And the answer is yes. So you can see here part of the brain of a fly, and you have here green, so that is the enhancer is active. And these cells are again early neuronal cells in the brain of Drosophila. So that tells us that from Nematostella to humans, there are some cells that have the same regulatory state, or very similar one. So in a way, we are telling that regulatory states are a great way of characterizing cell types. Right? Much more than just simply gene expression in general of some markers.
The three ABC figures in which this regulatory sequence is present, are they in some way related, which is different from the fly? I think, is it related in the... I don't think it's what you... So the three figures in humans and Amphioxus and sea urchins, I think it is. Yeah, we find these regulatory sequences and we find that it works in the fly, but it's not there in the fly. So can we find on the evolutionary chart some place where the fly was different from these three, or is it just completely coincidental?
No, what I think you could have found a number of combination of sequences, but in this particular case, it says that Drosophila doesn't use neither this sequence nor this particular gene, which is called SOX21, that is regulated normally by this enhancer. It's not used in the fly to make the brain.
So completely coincidental. It could be completely coincidental.
To be completely coincidental. Right?
So
yes, but okay, it could be completely coincidental if we had only analyzed Drosophila and other organisms, right? You might say, okay, I found that the human sequence in the zebrafish drives expression in neuroblasts, and I do the experiment in Drosophila, and it's expressed in neuroblasts. Okay, It might be coincidental. I mean, there are hundreds of cell types in Drosophila, but still, it might be a coincidence. But now, if you include all these sorts of animals in which you have a vertebrate, a sea urchin, and Drosophila, and you find that they are expressing similar cell types, now I would call them almost homologous cell types, then it's like the coincidence weakens. We believe that these truly recognizing the same regulatory state. But yeah, you are absolutely true. It might be, with some probability, it might be a coincidence. Okay, so now we are getting in the field to descriptions of how all the genes in the genome behave and how they are connected and how one transcription factor affects the expression of other genes and are becoming these sort of very complicated clouds of interactions. Here, I'm just simply give you an idea. These little balls here are genes, and the connections between genes indicate a regulation. And if the arrow is red, it means activation or repression, perhaps. And the blue ones is the opposite. Okay, so this is like a static representation of a dynamical system. Okay. So now the question that we have in the field is how do we go from here, right, to a tissue like a Drosophila retina, where you have cells of different types arranged in spatial, stereotypical ways. So how do we go from here to here? So one way of simplifying the problem is that within these networks of regulatory connections, yes? They refer to the activatory or repressing action of these interactions. So having this one would repress this other gene, having this one might be activating this other one, okay? So I don't have time to explain you how one gets to this sort of representation, but there are ways in which we believe that this is static representation of regulatory interactions. But anyway, it might be true or it might not be true fully, but the important thing here is that within the genome, these interactions of many like this are happening, right? So you have this cloud of interactions. So you need to sort out from this cloud of interaction how you go from here to here, to a real tissue. And I was telling you that there within this, what we are going to call now gene regulatory networks, there are some motifs that dominate the chemistry, right? Dominates the chemistry. So, and perhaps one way for those of you who are not familiar with these dominating reactions is to start like reading some reviews by René Thomas, who's one of these physicists who worked in Brussels slightly later than, for example, Ilya Prigogine was describing the dissipative systems away from the equilibrium and speculating about the nature of biological systems. So Brussels at the time was a really interesting place to work in biophysics, I assume. So René Thomas started trying to understand how these reactions can be controlling processes. So he started like saying, okay, if I have one gene, which is going to be like that, and it's activated for a while, if I initiate this expression and then I remove the stimulus, then gene expression is going to go down. So this will give rise to a pause. So what if I want to maintain the expression of the gene? One way in which you can maintain the expression of the gene is by making it autoregulatory. And we see these many times in transcription factors. Of course, that would lead to an exponential increase in transcription factor concentration, but this doesn't happen. Right? It saturates at some point. Why? Because this transcription factor, when it feeds on itself, it acts on a cis-regulatory element, and that cis-regulatory element can cope with certain amount of protein, no more than that. So okay. So, for example, these can work in growing domains. For example, now, and I'm already moving into developmental systems. So imagine that now you have a signal that activates a transcription factor called distallase. Again, it doesn't really matter the name, at the radius, right? So now you activate distallase, the green gene, in this whole domain. Now imagine that distallase is capable of auto-regulating. So now, if this domain grows, and it grows beyond the limit of the signal that initially induces distallase, still, the cells that come from this region and divide will maintain the expression of distallase. Okay? So that's a way in which you can mark a group of cells and then label them, even though they are growing. So you can scale, if you want, the expression of a gene just simply by making it autoregulatory. The autoregulation might happen directly or indirectly, right? But this is a nice way in which you can do that.
Here the growth is because cells are being born, new cells are being born.
Exactly. When you will see that in my slides next week. When I increase the size of a domain, it's because there are more cells, right? And it's exactly what happens. So a cell that was green here gave rise to two cells, one that remained here and one that remained beyond the reach of the signal. And that cell would be white otherwise. But since it contains distallase, it will auto-regulate its expression beyond this range. So you can start with small organs and then make them grow larger, and that would be enough. So yeah, I think that it would be a nice way of now to tell you something about scaling. We were discussing the other day about some instances of scaling. So, so the neural tube of vertebrates is something like that. And when we are small, we have a source of sonic hedgehog again. Sonic hedgehog is going to come up many times here. And sonic hedgehog will be a signal that will come to this region here and then will be produced here. And let's imagine that now we have sonic hedgehog inducing one fate, right? It's that fate one. And now you have here fate two, which is produced when these cells do not receive sonic hedgehog, right? Of course, if this thing grows, you're producing sonic hedgehog here, and sonic hedgehog only reaches here, now you will have fate one here and fate two here. So this would be disproportionate. This is not what happens. So initially, sonic hedgehog subdivides this population of cells. And once these cells have received a hedgehog, they in some way, they auto-regulate their fate. So when we grow up, this is what we get, right? So in many cases, scaling can be justified by an early patterning event that specifies domains, and then by scaling of that pattern, because there's auto-regulation within the genes that are expressed in the different domains. I mean, the thing is more complicated than that, but we can discuss in more detail how it happens, but basically this is the concept. So one thing that
so in this case, if the duplication is happening after sonic has penetrated this portion, then this pattern will be repeated. Will enlarge without change of
proportions.
Proportion, right? So it's whereas if sonic was to go in
constantly,
then it would not.
Exactly. Exactly. So if you want to know more about this, how this process happens, I would recommend you to go and read papers by James Briscoe. Okay, so there are recent reviews on this issue that you can read about. So one other thing that autoregulation might be able to achieve is to refine gene expression boundaries. Right? Because this autoregulation might actually be dependent non-linearly on some positional cue. So imagine that you are a cell that could be autoregulated here or here, but this loop depends not only on the presence of yourself, on distallase, but also on the presence of this factor, this orange factor. Basically what happens here, right? So once you have this thing here, only when there's the transcription factor plus this other signal, you would get some sort of step function happening here. So this also helps in refining gene expression boundaries. And here what I'm representing just simply length of the tissue. So here it would be the tissue you want. Another very interesting regulatory motif, and I'm talking here about very simple motifs, is the negative feedback or auto-repression. Right? And here you have a number of books or reviews where you can actually read more about not only the dynamic properties of these motifs, but also how they are used in biology. So this feedback auto-repression is a motif that, okay, as the gene product goes up, it will start like repressing itself. So it will lead to some sort of stabilization of gene expression. This repression will actually act to dampen this activation. Right? So when initially you activate it, but as you go up, you repress yourself, and these systems can actually stabilize gene expression. So initially, usually with time, the profiles that you see after activation are something like that. Right? You activate gene expression, it goes up and then stabilizes there. So these are ways that systems use to stabilize gene expression at specific levels. So if you go for whatever reason the system is noisy, and you go and increase the expression, it will go back again. If you go below these levels, it will go up again, right? Maintaining stable gene expression. I'm going to skip this one. And I will just give you another example. We've been discussing about this system. This is a system in which is based on this sort of motifs, right? In which cells can be either of one type or other type. Right? This is a motif called toggle switch, which works like a toggle switch. And basically, it allows the system to be either on one state or the other state. We've seen some examples even this morning. But of course, when you want to distribute these two states, A and B, in space, usually what you do is, again, use signaling cues. So imagine that this cell might be either of these two states with some probability. So if this cell is exposed to a signaling molecule, this signaling molecule might actually bias this decision, so making this reaction stronger than this reaction, and then therefore this cell will be A and not B. So this is a way in which cells actually, even though they can have potentially the possibility of being A or B, they get distributed. So A will be on one side and B will be on the other side. Right? And this is just achieved sometimes because they're exposed to some signals. So cells that are close to high levels of the signal will be biased to become purple, or because there's a cell nearby that is biasing you to actually take another fate. Basically, these biostable loops can be modified by the environment, and therefore you organize patterns with these sort of motifs. And I want to leave it with these two slides. We also mentioned something about eye development. I've been talking about genes and gene function and how genes are regulated and how many genes are regulated and how within these clouds of gene regulation there are some reactions that are more important than others, and some examples about, for example, how these motifs might regulate the behavior of the whole network. And my intention was to say that genes are modifiers of what cells do more than the genes direct cells towards one or other fate. But still, there are some examples in which we see the power of genes. So this is one example in which you have a number of eyes, the human, the mouse, and the fly. And there were a number of mutations identifying these three organisms, in Drosophila called eyeless, because of obvious reasons. In the mouse, it was called small eye, and in humans, it's called aniridia. So it happens that these syndromes in the human, in the mouse, which are small eyes, and in Drosophila, which is a small reduced eye, are produced by mutations in the same gene in the three species. And when I'm telling you that it's the same gene, it's a homologous gene. It's the gene most similar between species, indicating that the common ancestor of all these humans, flies, etc., had that gene. That gene is called, in general, it's called PAC6. It's a transcription factor, right? So this is a lateral view of a fly when it doesn't have eyelets, no eye. So now, what if we overexpress in different places of the fly eyelets artificially? So instead of, it's like we would evolve new regulatory regions for eyelets, and now instead of eyelets being expressed in the future eye, it's expressed in many other places, like in the antenna, in the legs, in the base of the wings. So what the expression of eyelets, or even the human version of eyelets does, is this. You start like getting eyes in the antennae, in the legs, or in the wings. So you can produce flies that basically have like, I don't know, like 12 eyes or 14 eyes on the body, just by manipulating the expression of a single transcription factor. So that indicates that there are some genes that are more important than others also. And this is one example. So I wanted to leave you with this.